personally salute all frontliners who have worked tirelessly and innovators who have yielded a solution to prevent and treat COVID-19 and other crisis situations, or an initiative which has contributed towards mitigating the impacts of coronavirus and providing relief to communities. I have observed abundant creativity and innovation emerging at the national, institutional, organizational, and individual levels for the past year.
Now is the ideal time for initiatives like this to be explored, not only encourage development of innovation, but to manage future emergencies and crises.
Sejak mencapai kemerdekaan, Malaysia telah mengalami transformasi yang pesat demi kemakmuran dan kesejahteraan rakyat. Sektor kesihatan, antara sektor terpenting negara, sentiasa berhadapan dengan pelbagai isu dan cabaran dalam era globalisasi ini. Fokus Kementerian Kesihatan menjadi lebih luas, terutamanya dalam menyediakan kemudahan kesihatan yang saksama, mudah diperolehi dan berkualiti. Untuk itu, perancangan dan pelaksanaan program kesihatan negara perlulah berasaskan kepada bukti saintifik menerusi hasil-hasil penyelidikan yang berkualiti. Penubuhan Institut Kesihatan Negara atau NIH yang bernaung di bawah Program Penyelidikan dan Sokongan Teknikal Kementerian Kesihatan Malaysia adalah bertujuan untuk menjalankan penyelidikan yang mana hasilnya dapat memberi bukti saintifik terhadap pembentukan polisi dan intervensi bidang kesihatan. NIH telah diluluskan di bawah rancangan Malaysia ke-7 dan secara rasmi beroperasi sejak bulan Ogos 2003. Visi NIH adalah untuk menjadi organisasi penyelidikan kesihatan terkemuka ke arah meningkatkan kesihatan dan kesejahteraan negara. Manakala misi NIH adalah menjalankan penyelidikan, latihan dan perundingan berkaitan kesihatan yang berkesan dan berimpak tinggi untuk meningkatkan kualiti hidup. Selain itu, NIH juga bertanggungjawab untuk mentadbir dan menguruskan penyelidikan di negara ini untuk menangani isu-isu kesihatan nasional. Kompleks NIH seluas 40 ekar di Setia Alam Selangor mempunyai ciri-ciri seni bina moden yang melambangkan kerancakan transformasi perkhidmatan kesihatan yang dizahirkan oleh penyelidikan yang berkualiti tinggi. Dilengkapi dengan pelbagai fasiliti serba moden, NIH turut menekankan kepentingan ekosistem alam sekitar yang mampu mewujudkan suasana kondusif untuk penyelidikan. Kompleks NIH menempatkan sebanyak 6 buah institut yang mempunyai pengkhususan dalam bidang penyelidikan masing-masing. Kelancaran operasi setiap institut ini disokong oleh pejabat pengurus dan pejabat pendaftar yang menjadi tunjang utama dalam pengurusan strategik dan pentadbiran NIH. Institut Penyelidikan Perubatan atau IMR merupakan institut penyelidikan biomedical tertua di Malaysia yang diwujudkan sejak tahun 1900 oleh Kerajaan British. Ketika itu, ia dikenali sebagai Institut Pathology di Kuala Lumpur. Hasil penyelidikan IMR telah banyak menyumbang kepada pengetahuan, pemahaman, rawatan dan kawalan beberapa penyakit tropika utama di negara ini dan rantau Asia Tenggara. Kini, fokus IMR juga kepada penyelidikan bioperubatan seiring dengan trend perubahan isu dan beban kesihatan negara serta kemajuan teknologi perubatan. Sejarah Institut Kesihatan Umum IKU bermula di Singapura sebagai Royal Sanitary Institute dan dijenamakan sebagai Royal Society for Promotion of Health RSH yang berperanan melatih inspektor kesihatan dari Tanah Melayu, Singapura dan Borneo. IKU berkembang menjadi pusat penyelidikan epidemiologi dan kesihatan awam yang menyediakan data-data kesihatan penting pembuat dasar dan pihak berkepentingan mengenai prevalen penyakit dan taraf kesihatan rakyat negara ini. Antaranya adalah tinjauan kebangsaan kesihatan dan morbidity at National Health Morbidity Survey yang dijalankan secara berkala. Institut Penyelidikan Klinikal atau ICR berfungsi sebagai cabang penyelidikan klinikal KKM dan mula beroperasi sejak Ogos 2000. Dengan 5 buah pusat kajian yang berperanan menggerakkan kajian-kajian klinikal tersendiri serta mempunyai 36 cawangan di hospital-hospital utama KKM di seluruh negara. ICR memainkan peranan penting dalam menerajui pembangunan dan pengukuhan kapasiti penyelidikan klinikal negara, mewujudkan dan mengekalkan pengkalan data klinikal bagi KKM dan juga mewujudkan kerjasama dengan organisasi penyelidikan tempatan, serantau dan antarabangsa mengenai penyelidikan klinikal. Institut Penyelidikan Sistem Kesihatan atau IHSR ditubuhkan pada November 2002 bagi membantu KKM dalam pelaksanaan penyelidikan terhadap sistem dan pengurusan kesihatan negara yang membantu dalam menyediakan bukti saintifik kepada pengubal dasar dan polisi kesihatan negara mengenai taraf dan kualiti penyampaian perkhidmatan kesihatan kepada rakyat. Institut ini berperanan dalam melaksanakan penyelidikan berimpak tinggi dalam bidang penyampaian perkhidmatan kesihatan negara, melaksanakan penilaian terhadap dasar dan polisi kesihatan dan kebolehcapaian perkhidmatan yang saksama dan berkualiti dalam kalangan semua rakyat negara ini. IHSR telah mendapat pengiktirafan antarabangsa dan telah dilantik sebagai Pusat Kerjasama Serantau WHO untuk peningkatan kualiti dan kesihatan. Institut Pengurusan Kesihatan IHM bermula di bawah Institut Kesihatan Awam yang dikenali sebagai bahagian latihan pengurusan. Penubuhan IHM bermula pada rancangan Malaysia ke-6 yang mana ia bertanggungjawab dalam meningkatkan pembangunan modal insan profesional di KKM untuk menghadapi cabaran dalam penyampaian perkhidmatan kesihatan yang berkualiti. Bidang penyelidikan yang dipelopori oleh IHM ialah aspek kepuasan pelanggan, pengurusan sumber manusia serta pemantauan dan penilaian terhadap kualiti perkhidmatan kesihatan negara. Selain itu, IHM turut berperanan sebagai sebuah institut yang menyediakan latihan berkualiti tinggi kepada kaki tangan KKM. Institut Penyelidikan Tingkah Laku Kesihatan IPTK ditubuhkan pada 2005 adalah bukti bahawa bidang tingkah laku kesihatan adalah penting dalam usaha mencapai sebuah negara maju. IPTK bertanggungjawab dalam melaksanakan penyelidikan inovatif dalam bidang tingkah laku, penilaian intervensi promosi kesihatan, kaedah komunikasi kesihatan yang efektif dan komunikasi risiko. Penyelidikan yang dilaksanakan oleh penyelidik-penyelidik di NIH bukan sahaja telah memperbaiki penyampaian kesihatan kepada rakyat Malaysia, namun telah turut mendapat pengiktirafan di dalam negara dan di peringkat antarabangsa. Way Forward NIH akan terus mengkaji dan meningkatkan kepakaran para penyelidik ke arah menghasilkan kajian-kajian berkualiti dan berimpak tinggi dalam meningkatkan taraf kesihatan dan kehidupan rakyat Malaysia.
Ladies and gentlemen, our next lined up program is a knowledge sharing session. Please enjoy.
Thank you, organizer, for the kind introduction. First of all, I would like to thank the organizer of Malaysian Technology Expo 2022 for this opportunity to introduce our work on pharmaceutical technology. I'm a representative from the Malaysian Society of Pharmaceutical Technology, a professional body for pharmaceutical scientists in the country Malaysia. And today I would like to share with you a little bit about our work, the electrospin of pharmaceutical active into next generation dosage form, the potential and the opportunities. So whenever we talk about this pharmaceutical dosage form, we have to first understand who are the persons that is going to use this dosage form. So let us have an overview. It will be not a one-size-fits-all pharmaceutical dosage form, that we need to recognize the particular target group. For example, the elderly, those who are having a chronic disease, the children who are having disease, those with high risk and vulnerables, and the rare disease such as cancer, rare disease from the certain populations, and of course the public, and particularly women with the childbearing age, pregnancy, and also women's sickness. By having in mind that those are the population that we are targeting, in each design of these formulations, we can then cater to their needs. So a brief overview about this drugs discovery pipeline. It takes over 10 to 15 years within an average cost of 1 to 2 billion for each new drugs to be approved for the clinical use at the end. Okay, so when we look at the pipeline, the beginning will be actually finding a target that actually can work on our human bodies. So that will be the molecular matching, and it was found that only 10% of the human proteins are known to be druggables. And from this 10%, when they move on to this high throughput screening using the characteristic of the human receptor, and it was found that more than 90% of this new chemical entity are reported with poor biopharmaceutical properties. This is where we need drugs development. And in the drug development phase, actually, when we have done this in vitro study, less than 12% of them is approved for clinical trial. So this is because there are a lot of failure in terms of the in vitro designs and things like that. And subsequent to this, we have to do the safety, efficacy, and only less than 5% of those remaining will be approved and be used by the humans. So when we talk about the failure in the drug development, the perspective of where and how we are going to tackle this problem is actually matters. So let's say we look at the raw material. The raw material is not only the new chemical entities that I have just presented in the previous slide. It is also excipient, the carrier, the polymers. And what are the strategy that we are trying to do when we try to develop the drug development process into a pharmaceutical dosage? Different strategies such as complexation, salt formation, nanotechnology, advanced material, solid dispersion, liposome, and etc. can actually be utilized as a strategy. And then they are being manufactured using, let's say, spray dry, freeze dry, electrospinning, only then subsequently to a final dosage from processing, which is tablet, capsule, elixir, emulsion, patch, and etc. With all this strategy have gone through from the raw material to final dosage form, all the final dosage form look the same. Okay, they look like tablets, they look like a capsule, injection, and etc. But if you look at the products here that I've shown, for example, Sporanox, they are actually utilizing the strategy of solid dispersion, Prograph, strategy of solid dispersions, doxorubicin vials, which is using the strategy of liposomal, and etc. So those are things to be bear in mind. They are not all simply put in the powder and kept in the tablet, and that's all. So they are with the strategy, then only go to the manufacturing tabletings and capsuling. So let us talk a little bit about currently what happened with this current manufacturing method, which is the spray dry, freeze dry, and recently electrospinning has been emerging. And electrospinning in our country is actually available, but more onto this material production rather than pharmaceutical dosage form. So it has a vast potential in the pharmaceutical dosage form. So let's look at the principle of electrospinning. You need a high voltage source to inject your solution. And when you need to inject your solution will be in the syringe with a polymer solution with the drugs, and of course a collector, okay, both being connected through this voltage. And once the electrical difference has been generated, then the syringe pump from the other end will be pushed slightly, and this excessive solution from the tip of this syringe will be charged, and then because of the grounded screen, and it will travel to the grounded screen, and along the journey the evaporation of solvent occur. So in brief, the process of electrospinning, they generate electricity field, charge repulsion causing the solvent evaporation simultaneously happen and accelerated to be the grounded collector, and sample being collected, and you can proceed to production and packaging. So electrospinning, when you look at the previous diagram, it was actually in the form of a fiber, and this fiber can be down to the nanoscale. However, it can also to be formulated into particles. Okay, depending on the type of the viscosity that you use for the solution, it can be a particle, it can be a fibrous. So this is just a very quick overview about the electrospinning process in the lab that you can see. So what are the potential of electrospinning? Let me talk you through a little bit about the journey that I have walked through across electrospinning. I have started my electrospinning experience in the UK, where I electrospin zinc, okay, a type of protein. And we have found that nanofiber range was actually found, and the addition of any spin substance can causing beaded architecture of the fiber. When they are not miscible, they can cause a lot of beaded architectures. And soon after my UK visiting researching scheme, I have joined Professor Yuan on the electrospinning on production of nanofiber and also micro particles. And in this lab, we have utilizing this polyvinyl alcohol as our base. And when we are measuring the tip to the collector distance increase, okay, you will cause the fiber, the diameter of the fiber decreasing. And if you increase the voltage, again, the diameter can be decreased. And of course, when you have the viscosity increase, then you have a fiber with a more consistent diameter. And in this group, we are actually trying to electrospin probiotics that are producing nanofibers. And if you can see here, this is the forest particles is actually the encapsulated probiotics. So soon later, I correspond to one of my own works on the electrospinning, which is a buccal delivery of atorvastatin, a type of anti-malaria drugs, where we use a solid dispersion technique. And this is initiated by Miss Teo, which is one of my PhD students. We are actually looking at designing this anti-malaria patch, okay, with the limited amount of water, because when we have the nanofiber created, and because of the high surface area, when you put into your mouth, it can be oral dispersible with only a very limited amount of fluid in your oral cavity. It can be as small as just the saliva. And what has been done by Ms. Teo has been shown that when you have a physical mixture, a simple mix, that you will find that the release of the drug is very, very minimum. And when we electrospin them, and this is what we have seen. And because of the parachuting, patterns of the drug release, it is a typical representation of the solid dispersion dosage form. And we also play with the different type of polymer. We have PVPVA as well as PVPK90. It was interesting to find that when we use PVPVA, we got a particles powder rather than a fibers. And when we increase the ratio into the PVPK90, which is the viscosity has been increasing, and we found that our fiber becoming more and more consistent from C, D, E, and F. And of course, this journey was humbly recognized as a National Pharmacy Research Competition as gold. And of course, we win this KPJ Healthcare Conference award, and also Asian Young Pharmacy Group Summit as a winner for this product that's been presented in this summit. And of course, I have also collaborated with another group, which we are electrospinning the nanocellulose. And as this nanocellulose was actually not dissolved in the solutions, so we are attempting to electrospin suspensions. And when we electrospin suspension, the needle gauge is very critical. However, viscosity remained the cornerstone for the formations of the sustainable electrospun. And we have found that with increase of the NCC, the film strength increased, and we can able to generate a control release dosage form. Okay, this is Mr. Salam who are working on this. I have also a collaboration with our local private university on the shift-based complex for anti-cancer, and we have successfully formulated into the amorphous solid dispersions, as can shown in the X-ray diffractions. And if you can see, the dissolution has improved dramatically, more than 20%. And one of the very recent works that we are working on is actually electrospinning of emulsions. So why is it called emulsion? Because of the incorporated substance that in our solution we're using red palm oil, one of the local resources, and we are actually developing it for children's supplementations. And once again, oral dispersible for children, and the populations of the target is being designed in such a way that children have difficulty in swallowing. So having this design will be actually enable and facilitate this compliance problem among the children populations. So this is Mr. Kong that he has been working on investigating the parameters. So they are actually looking at the viscosity as well as the conductivity and also other solution property to actually get into a good peelable film. Okay, and if they are not peelable, they are not acceptable for finance formulations. And of course, viscosity study, and we have found that additional of plasticizer can increase the elasticity of the film. However, it may actually reduce the disintegration of the film. In this particular works, once again, we have proved that viscosity play a very critical role in this electrospinning film fiber. However, we have got a very interesting observations. While we are trying to electrospin emulsion, tree-like structure like this fiber will actually seen when after we have spin them for more than 2 hours. Okay, after we have done this, we have gone through and see what is actually this tree-like structure, and we have just realized when you electrospin an emulsion stability is one of the very important characteristics. So we have to make sure that the emulsion is highly stable. Then we go for the electrospinning, and the electrospinning time frame shall be limited to the stability of the emulsions. Okay, those are some of the picture that I can share. So we call it ramified jet. Okay, and on the similar works on this oil, we also encapsulate the oil with another type of excipient by Mr. Huang, that he has won the award under the National Pharmaceutical competitions against, and he has successfully shown that we are able to encapsulate the oil within the fiber, that the excipient that he used, and the diameter of the fiber increased with the increase of the amount of the oil. Of course, the threshold of where the oil can be incorporated is one of the very interesting parameters that we are currently still working on. So this is some of the example that you can see that when we prepare a peelable film, and this is what can be directly cut into the desired size and packed into the final dosage form without much of the processing. And I have also one of the researcher, Ms. Tan, is actually working on coaxial accessory. Coaxial accessory means that the electrospinning, still same needle, but this needle have 2 layers. So this is the example of the needle that you are seeing. So you have the internal face of the needle, it's like one metal needle, and another metal was actually covering this needle, and they have 2 inlet of the point for this accessory, and the 2 inlet can actually insert 2 different material into the respective, the core and also the shell. And if you have anything that is highly degradable, you can actually put in the core, and you can actually protect it with the shell. And this is what we have worked on, the papaya leaf extract. Papaya leaf extract, as suggested by the IMR, to be processed under a cold conditions, okay? Because the bioactive is either the alkaloid or either this flavonoid, and one of them is actually thermosensitive. So to make it remain as active, so it was not recommended to actually boil the leaf, but simply with the juice of the leaf. So this is what Ms. Tan has been working on, just to protect this papaya leaf extract and get into straight to the final dosage form without much of the processing that can cause possible degradations. So this is one of the works and the potential that what electrospinning can actually offer. So I have run through a different strategies and the dosage form that can be produced by the electrospinning. We have gone through solid solutions. I have mentioned solid suspension by the nanocellulose, solid emulsion with the incorporation of the oil of the red palm oil, and of course solid dispersion by Ms. Teo on the anti-malaria drugs. And all of this, you can have them in the powder form that you would like to further process them in tablets and capsule. But you can also have a type of powder form, a type of dosage form, which is film, that could be directly cut into the desired size and pack. So this is when the strategy and the dosage form has met into a very simplified manner. Okay, so what are the opportunity to bring this electrospinning further? Fundamental research actually is needed, because in this aspect, in this country, this is something rather new, and support from the company as well as the company to buy in the idea and the scale up to put in the good manufacturing environment is really important to make this forwards. And of course, regulatory needs in approving the registration of products such as this, because a film can either be determined as the pharmaceutical products, or they can either be registered as just a food. So this is the category that needs to be redefined in the regulatory aspect. And of course, the participation from the patients to actually tell us their preference, and the market survey about the acceptance about the children as well as the geriatrics will be a very variable data to be shown. So in order to realize the potential, once again, I suggest that if we have a fundamental research funded with scale up possibility, market survey, GMP and regulatory, then we will have those kind of very advanced next generation dosage form appear in the market for the benefit of our society. So now I just show you some products that has been patented by using electrospinning. Okay, most of them, such as face masks or surgical implant, okay, or wound dressing. For any oral drugs or pharmaceutical products, we have these patches, okay, that is can be found on the market, but very limited products. So in conclusion, electrospinning can be applied to different strategies: suspension, solution, probiotic, emulsion, new chemical entity, etc. It can be directly manufactured into final dosage form, which is a film, or intermediate, which is powder or even patch. Okay, it will generate products with high surface to volume ratio, And because of this fiber diameter that you can actually tune by using the viscosity of the solution, you can actually tune the porosity that you would like to have depending on the solution compositions with the various size and shapes. And it is easy to control with the desired functionality. Just a very quick overview about my collaborator now. We are moving forwards to the larger scale. I have tried a different type of electrospinning. We have multi-needle. We have also needleless to fast and upscale the productivity. And nationally, I have my collaborator from IMU, from PowerLife, Progene, MPOB, and also some local private university. And internationally, of course, I have my collaborator in Italy, in Iraq, and also in the UK. And I would like to thank my mentor, which is Professor Emeritus Yuan, and also my supervisor, Professor Duncan. And of course, my school with the endless support of the facilities and facilitating the process of applying any funding that is given to me. Thank you very much. Of course, not last but not least, I would like to thank also my lab mates. And here are my lab mates, or you can say it's my student, but I always take my student as my lab mates. And thank you very much.